Hello everyone. In this session we are going to talk about biochemical oxygen demand. Which are the main parameters for measuring water quality? In the last class we studied chemical and physical characteristics, and some parameters were studied in the 70s are water quality index, that compare pollution levels in different areas, and we have in here the parameters. Some of them are physical parameters like color, pH, suspended solids, or dissolved solids. If you see these parameters, we have some derivatives from the textile industry, like active substances like blue methylene and some dyes. The current parameters are equal, important, and we have in here some common aspects like biochemical oxygen demand and the total suspended solids. We have in here the organic matter decomposition, and if you see in here, this cloud represents the organic matter, and we have the bacteria in here. This organic matter is degraded by the bacteria in some more simple substances. This degradation could be aerobic or anaerobic, and we have in here in the form of a chemical equation the organic matter in presence of oxygen is degraded by these microorganisms, and the products are carbon dioxide and water. These main products are also obtained by anaerobic degradation, but you see here that it is not oxygen. Then the subproducts are the difference. We have noble cells, but the side products obtained with aerobic degradation are stable, like these ones. These ions obtained in the effluents of water, you can see that there is no problem because they are stable products. But in here in anaerobic degradation we have unstable products like some gases. There are combustible gases, and this is the problem. Then we have to obtain some effluents that have oxygen dissolved in the water. The BOD is used in many analysis from the industry, and we have in here the definition from the the NMX normative, that is the estimation of the amount of oxygen required by heterogeneous microbial pollution to oxide organic matter of a water sample in a period of 5 days. This standard is used in many countries. 5 days to obtain this measure. If you see a quantitative estimation of the amount of oxygen to degrade that the bacteria or the microorganisms could degrade organic matter. So we have in here an example of the analysis, analytical analysis. We have in here this first bottle that is a diluted bacterial culture, and we have in here the bacteria in distilled water and the essential nutrients. So we have in here a culture, and from here we separate in 2 bottles, that one is the reference and the other is mixed with the sample of wastewater. So we have in here the flask with the sample diluted in with bacterial culture. Then we have two types of measurements of the biochemical oxygen demand. One is the estimation these 5 days without the wastewater sample there directly, and this other option that is using a bacterial culture to measurement the BOD. An important data is that the flask to measure this the quantity of oxygen remaining in the samples is that the flasks are of 300 milliliters. This is a standard also. Then, what is the water quality according to the BOD 5? We have this in Mexico, this color for the water quality from blue to red, that in the standard we have the BOD 5 less than 3, then we have a non-contaminated water. If we have a BOD 5 up to 120, we have a contaminated water. More than that is strongly contaminated. And you have in here this table that says the water quality according to the BOD. This is only one parameter, so it's an indication if the water is contaminated or not, but we have to use more parameters when the water is meant to be purified water to consume. How can we calculate BOD of a diluted sample? We are using this formula. You can see in here DO. This means dissolved oxygen, and we have an initial dissolved oxygen minus a final dissolved oxygen, and this P is the dissolution rate, that is the wastewater volume in the wastewater volume plus the dissolution water. You can see that this, if you remember the bottles, you can see this volume is the total volume. So is the dissolution rate is a fraction of the water volume. The water volume in the total volume of the sample. So in this class we studied about biochemical oxygen demand, and we determined two types of BOD, which is one without bacteria, and the other one with culture solution. So there are two ways to measure this analytic parameter for obtaining water quality in wastewater. Thank you.